So I'll try to tell you some stories about soft and biological matter. So we call this living matter, but I'll try to focus on some simple statistical physics aspects of living matter and try to put out some ideas. So what I'll do is the first half of this talk, or maybe 3/4 of this talk, will be more pedagogical. So I'll try to put out the philosophy of thinking about living systems, perspective of statistical and condensed matter physics. And then towards the halfway through or something like that, I'll go to the board and I'll work out some problems in diffusion and also some problems in stochasticity that we were discussing about in the last talk. And so that will be the sort of pedagogical part. And towards the end, I'll try to put an emphasis on the kind of things that I do. And tomorrow, I'll really show you some of the results that we work on. So I want to start very broadly. I mean, it might sound a bit too big, but let's just bear with me. So when I want to think about matter and perspective, so there are basically two big questions about the world. What's the world made of? One is you try to understand structure at various levels, from atoms and maybe something beneath, and you go on across molecular, structural scales, human, planet, sun, and so on and so on. Okay, so that's one description of trying to find out what is at which level and what is the structure. The other question is, how is the world put together? So that's a much harder question, much harder question. So what do you mean by that? I want to understand the following. I'll give you nucleons. Can you build me a nucleus? I'll give you protons and electrons. Build me an atom. I'll give you lots of molecules. Build me a solid. And so on and so forth. So this idea of putting together things and trying to see the emergent properties of large-scale aggregates is a pretty hard problem. So we had such discussions last week, Olivier, the two Oliviers, both of them talked about statistical physics approaches to many particle systems. So we see that, right? So if you just put in a large number of them together, they exhibit very non-trivial properties. So what do you mean by non-trivial? These are not properties that the individual entities have. These are really properties which are an attribute of the entire collection of objects. The standard and very dramatic thing is what is called phase transitions. I'll discuss this in more detail. So think of electrons in a metal, for example, the superconductivity that we think about. A single electron is not superconducting. It's really an aggregate. It's really a property of the entire collection put together that gives you these properties. Very non-trivial. In biology too, just because we understand something here doesn't mean that we understand this. Because as soon as you put a large number of them together, there are certain really complicated behaviors that come up which cannot be explained by just looking at individual parts and putting them together. It's really properties that exist for the collective whole. Those are the kind of things that I really would like to understand. So Olivier last week told us about emergent properties of complex systems. Right? So these are really properties which exist only for interacting many body systems. Think of standard elasticity, viscosity, conductivity. There's no viscosity for a single molecule of water. There's viscosity only when you put a large number of molecules of water. Right? So there's really a property that comes up when you put a large number of them and interact with each other. So one would like to ask the question, suppose I know the properties at the basic level, is the properties at the higher level just the sum of them, or they're really interacting and so therefore they're very non-trivial properties that can come up? I'll go very slowly. So the way we think about these things in statistical physics is that if you take a system at equilibrium, thermodynamic equilibrium, the way you do that is you take a system, just leave it there and just leave it for a very long time. So it will go to equilibrium. It's those kind of systems we know to describe pretty well. So if you have a system like that, let's say you have a region in which there are a bunch of particles, they might be interacting and they may not be interacting. We know to do what is called the statistical mechanics of these ensembles. So Olivier showed us how to write the partition function, for example. We forgot. The sum is over all possible states of the system. So if you give me the energy of the system and there is a temperature T, I can write down what is called a partition function. And the partition function from that, the whole of thermodynamics, free energy, and so on and so forth follows. And these large-scale properties of the system can be calculated, at least in principle. In practice, it might be a harder problem. You could also go to a situation where the number of particles, for example, is not conserved. So let's think of a cell, for instance, has some molecules which are sitting inside, but there are also the same molecules which are outside, and there are ion channels which can pump things in and out. It's a very simplified description, but one has pictures for these things if the system is at equilibrium. That's very important, only if the system is at equilibrium. If we don't have descriptions, if the system is out of equilibrium. But within an equilibrium context, we can still try to understand how is it that if you put a large number of them together, new properties come up. Most drastic is what is called phase transitions, right? So materials can exist in different phases of matter. Remember, the chemistry and the interaction between the constituent molecules is the same in all the three phases. They're not different. It's the same set of molecules, but you have put them differently. Put together differently. That's the key word. So we know how to go around, right? So we can take a substance which is at a solid, you can just heat it up, it goes into a liquid phase, and then finally it goes into a gaseous phase. So we know how to do this. It's a simple kitchen experiment that we do. So you look at the molecules of water. Surely you didn't change the chemistry of the molecules of water. It's just you supplied more and more energy on the average to the molecules, and they sort of assemble themselves in very different ways. And this assembly really leads to the emergence of new properties. What do you mean by that? Structurally, we know that in a solid, the things are nicely ordered, packed crystalline phases. Liquid, the order is destroyed, but there is some short-range ordering. On the whole, there's no average ordering like a crystal. Gaseous phase is less dense, and the molecules are really far apart and interact very often. Not very often. So how do we characterize this ordering? One way to think about it is the following. Let's say I have a bunch of particles put together, and I want to understand, is this in an ordered state like a crystal or a disordered state like a gas? So in the language of the Ising model, for example, you could think of it like a high temperature phase when the spins are all disordered, or a low temperature phase when the spins are all ordered and point in the same direction. One way to think in terms of a structural transition, let's say you try to calculate the pairwise distances. Look at all the particles. Look at the pairwise distances between all the particles. If you have n particles, you have n choose 2 pairs. So look at all the distances between them, just the distance, and calculate the distribution of the distances. This sort of gives you a very good idea about the ordering in the system. So if you take that pair distribution function and think about a vapor, gaseous phase. Surely the molecules are not interacting with each other. They just bump into each other very rarely. So that means if I have a particle here, and I look at the distance between this and the another particle, and look at the distribution of the distance, surely the two particles can't go into each other. So there the probability will be good. But otherwise, it's equally likely to find any of the particle anywhere. That means it will be a flat distribution beyond a certain distance, which is characteristic of the size of the particle. So if you go beyond the size of the particles, you're equally likely to find the particles anywhere. What about a liquid? Well, it's still disordered, but if you actually look at the density is higher, and if you really look at the liquid, there is short-range ordering. So it's not that you can find, if you have one molecule of a liquid here, you can find another molecule anywhere around. There are higher probabilities. So if you really look at that, you start developing structure. So surely they can't go through even in the liquid, but there are peaks that come up. So these are like our solvation shells. These are nearest neighbor shells that come up. The drastic version of this is a crystal. In a crystal, you only have an atom here, which means that really almost always surely dictates the position of another atom. Because it's a crystalline thing. So you know that the distances can't be anything arbitrary, but they only can be in some regularly placed positions. So there is a way to characterize ordering, structure in the system. But like I said, I mean, all this came about just by the same kind of interaction between the molecules, right? I mean, you didn't change the interaction. What you changed was some external parameters like temperature, pressure, and so on. So how do we rationalize this? So let's say you think about some typical energetic interaction between two molecules. So let's say there are two neutral atoms or molecules, and so they're not charged. They're neutral. So what will be the interaction between them? Forget gravity for the moment. Gravity is negligible in this case. So there's no charge on the hole. On the hole, there's no charge. So will they attract? Will they repel? They won't do anything. There will be a hardcore interaction. Surely the particles can't go through here. But what about if you separate them beyond their overlapping distance? So if you really look at these simple calculations, and you can rationalize this from simple chemistry ideas. Idea is the following. Surely you can't push them inside each other. There's a hardcore repulsion. That everybody understands. It's like a nuclear repulsion that's going on. But if you put them far away, there are instantaneous dipole-like moments, van der Waals forces that we think about in chemistry, right? That leads to a weak but attractive force. It's a long-range, weak but attractive force. So there is an attractive component to the interaction, and there's a repulsive component to the interaction. And somewhere they balance, and that's where there's a minimum in the potential. How do you understand phases from here? This is a typical potential between neutral atoms. You can take any of them. The details might be different, but the general structure. So that's what I think. Now, there's another energy scale in the system, the temperature, the control that you do, how much of energy that you put on the average into the particles of the system. So if the temperature is much higher than this potential, let's say the U0 is the depth well. If the temperature is much, much higher, the particles hardly look at the interaction which is attractive. They'll all just repel from each other, and so they'll bump into each other and go around. So therefore, they will be at this scale, the energy is, the interaction energy is much, much smaller compared to the thermal energy, and that's when you get a gas. So there's only a kinetic energy, there's hardly any potential energy interaction. If your temperature is comparable to the interaction energy, so that means they can't go through each other, but every now and then they do feel the attractive interaction, so they get trapped locally. Not long range, but locally. That's when you get a liquid. And if the temperature is really small compared to the depth of the potential, then surely if your particle gets trapped in the potential of the other, it can't go out. So then it's stuck. And so the next guy comes and thinks, so if you were there in Zorana's talk yesterday, they self-assemble. Right? And that's when you get a solid. So there's a way to understand from energetics how structure comes about. Now, if you look at the structure, you see these regular things in a crystal and random disordered things in a fluid. Surely, I mean, if you just catch hold of the molecules, if I catch hold of one of the molecules, is it stationary? Is it at the same place? I mean, there's a picture that I had, but if you look at a gas molecule in this room, for example, is it stationary at one point? Is it moving around? It's moving around a lot. What about a liquid? You take a glass of water, look at the molecules in the glass of water. Is it moving around or is it stationary? It's moving around. What about a solid? They don't move around? Surely not. Otherwise, it's not a solid anymore. But do they don't move at all? They do. I mean, they jiggle around in their mean positions on the average. That's what is temperature, essentially. So how do we characterize them? So look at some typical trajectories of the molecules. So if you look at the gas, they just move in straight lines. Hardly any collisions. Every so rarely, just the other guy comes and bumps. And so it's mostly straight lines, tapered with some small perturbation. What about a liquid? Well, it also does. But the amount of time it spends just going straight is very small because there are the higher density of molecules, they collide around, they move around, and they get trapped in that potential. Solids, on the other hand, will just be stuck on the mean position. They're really in the depth of the potential. They can't move around. They jiggle around a little bit here and there. So way to characterize this is what is called the mean square displacement. I'm sure you all do this. You track particles or cells or molecules. You calculate the mean square displacement. And most often you say you fit it to a straight line, r square versus t, and the slope is the diffusion coefficient. Where does that come from? Let's try to rationalize it. So let's say you look at the particle positions at some times t1, t2, t0, and so on. So look at the mean. So on the average, the particles go nowhere. So the mean is 0. But if you look at the mean square, that's MSD, and that's the number which I call r square. And let's try to ask how that depends, how that function looks like as a function of time, depending on which phase you are in. So look at the plot. So if you're looking at the gas, it's mostly moving straight. So position is linearly increasing with time, which means r square is increasing quadratically with time. So you get a graph which goes quadratically with time. What about a liquid? A liquid also does a quadratic increase at short times. This is the time it moves straight before bumping into the other molecules. But after that, the other molecular interactions come in, and that's where diffusion starts. And so therefore, the slope of the graph changes from being, the exponent changes from being 2 to being 1. So this is what you call diffusion. Can you guess what it will be for a solid? So at short times, will it still be like a parabola? Yes? No? Yes? Okay. Long times, what will happen? Will it go like T2? Will it go like T1? Will it saturate? Will it not go in? Huh? T half. That's a good guess, so it should go down. Anything else? It's a constant. So basically, it just goes to a constant. Why? Well, on the average, the molecules can jiggle around. But after some point, it can't really leave its mean position. It just becomes a constant. So if you look at a mean square trajectory, you can tell about the surrounding structure in the medium. That's a nice way, you know? So typically, when you see this, I mean, most biology experiments, we do it in solid, it's a liquid-like feature. So you say, assume it's t to the 1. It need not be. So that's the summary I wanted to give you. So the way we think about in equilibrium statistical mechanics is that if you have energetics, think of a typical interaction between atoms and see where the temperature scale lies. If the temperature is subdominant, you get a solid. If it's comparable to the depth of the potential, you get a liquid. If it's much, much higher, you get a solid. From here, you can rationalize the particular structure that is there in the system. It's basically the way it's ordered, disordered, the very order, short range order, and so on. And that really correlates with the thermodynamics that we know by actually doing experiments in the laboratory. We've changed atmospheric pressure or temperature and the various phase transitions that happen between these. Not only that, there's also dynamics. So there's energetics, there's a structure, the dynamics of the individual molecules. All this are related at equilibrium. If your system is at equilibrium, you get one, you more or less got everything. Not true if the system is not at equilibrium. And living cells are definitely not at equilibrium. So if you measure one and say something, you can't say about the structure in general. All right. So I just talked about isotropic objects, right? So if you just put them together, this is how the emergent properties come about. So I'm not saying I'm going to give you a derivation of these things. I'm just saying the way you put them together, a crystalline structure has elastic properties, a liquid structure has shear viscosity, a gas has nothing, it just has some compressibility. So these are not properties of the individual molecules, even though the interactions are the same. The way you put them together gives rise to these emergent properties. Soft matter is a definition we apply to material systems where the thermal energy is comparable to the interaction energy. And biological matter is soft matter because it's KBT that really makes the deformation of the molecules. Now, I just talked about isotropic objects, right? But there can also be more complicated things. So, for example, if you have the same liquid-like structure, for instance, of molecules, these are individual molecules that I talked to you about. But you could also construct polymers. That is, you could make a particular configuration of bonds between the objects and can string up a one-dimensional object, which we could traditionally call a polymer. You could have a polymer, and there are plenty of examples, DNA, proteins, so on and so forth. Lots of examples. You could also take the same system, but string it up in a different manner by putting bonds. So you can get a two-dimensional object. It's really a membrane, the thing that surrounds the cell and gives a nice compartmental structure. Right? So the continuum limit, you're basically looking at curves and surfaces. So one can do the statistical mechanics of points, which is what we did earlier. You could also do the statistical mechanics of curves or the statistical mechanics of surfaces, and so on and so forth. Again, equilibrium, we know a lot about these properties. You can also have something more non-trivial, what are called liquid crystalline structure. So individual entities that I talked about were points. What if they were not points? They were really extended objects which had some directionality to the system. So let's say this object. This is not a point. This has one axis which is long and the other axis which is short. So these are really anisotropic. So for instance, if you had rods, there are other kinds of transitions possible. So I'll show you two snapshots, exactly the same way the center of mass of the objects are oriented. So you would just say, oh, these are both liquids. But suppose it was actually rods and these were the center of mass position. What do you mean by that? Suppose I had the rods and they were all oriented in a random fashion. So it's liquid as far as the center of mass positions is concerned, but it's also a disordered liquid as far as the orientations are concerned. But you could have a transition where it's still a liquid as far as the center of mass is concerned, but as ordered with respect to the orientations. So you get a crystalline ordering in the orientation, but a liquid-like ordering in the positions. So I call it liquid crystal. Why is this important? Well, okay, you can find everyday examples of that too, but that's not the reason why it's important. It's also important, but you see that kind of structures in living systems. Think of the structures of microtubules that you saw in Olivier's talk last week. The microtubules were ordered. They are not point objects, right? They're really rod-like objects. Put a bunch of rods together and you crank up the interaction energy or lower the temperature. They will spontaneously order. And the ordered structures have different mechanical properties compared to the non-ordered structure. So the way they respond to any applied stress or strain is very different. Because if you apply a stress like this, they will flow like a liquid. But if you apply a stress like this, they will resist like a solid. So solid or liquid could also depend on what are you doing to the system. I was saying they could also form higher order structures called smectics. So here is a system where there is positional ordering in this direction because they are stacked. There is no ordering in the XY plane, they are randomly placed. But there is orientational ordering in this direction. I'll leave it to you to whack out your brains and think, if I shear this object, which directions is it a liquid, which directions is it a solid, and you could also have other kinds of anisotropic objects. Not rods, but disks. Disks are also anisotropic, right? So you could have stackings of disks. These are called discotic objects. Did people have idlis last week sometime? Yes, no? Okay. So you can take a bunch of idlis and make a discotic face. So maybe try that next time and see if it's stable. So what I wanted to impress upon you is the following. So these are phases of equilibrium systems. And so there we know how to characterize things about what phase it is in. And once we know the phase, we know how to think about the mechanical properties of the object, what kind of ordering comes up and so on. What about living systems? What do you think? Are we in a gaseous phase? Let's try to rule out one by one. Surely not. Liquid phase? Yes, no? Yes, no? Say something. Yeah? Okay. Solid? No? We're not solid? If I was completely a liquid, I probably wouldn't be able to stand here. I would just flow off. Why is it I'm able to stand and resist and retain the shape? How about the following? Let's do some examples. So these are what are called more complex objects, and we'll come to that. Suppose you take your hand, and I just pull my tissue like this, and I leave it. Snaps back. So is that like a solid-like behavior or liquid-like behavior? It's like a solid-like behavior. A piece of rubber, you stretch it and you leave it and it comes back immediately. The same thing if you take a glass of water, shear it, well, it'll just flow. So I take this, I hold it for a very long time, say 2 weeks, and then I leave it. Do you think it'll go back? Huh? Slowly, yeah. So that immediately asks the question, slow compared to what? So I didn't discuss this. There are materials where I talked about something being a solid in one direction, something being a liquid in other direction. There are also materials which are solid on short time scales, liquids on long time scales. Or the other way around, solids on solids on long time scales and liquids on short time scales. So what are called viscoelastic materials. This is an example of a viscoelastic material. So there's a certain time scale associated with cell division, cell rearrangement, and so on and so forth. If I pull it and leave it before this time scale, it will really snap back like a solid. But if I pull it and hold it and give it enough time for the cells to reorient, their positions to change and their morphology to change, the new position will come up, right? This will become your new hand with this deformation. So there will be an elasticity with respect to the new configuration. It will not snap back. Let's try to understand. I just wanted to give you a flavor for the kind of phases that exist and trying to understand what kind of phases they exist in living systems. Probably it's neither. It's neither a solid nor a liquid, and it's really a mixture of all kinds of phases put together. And that's where the complexity starts building up. We know to do the statistical physics of 1 phases. Okay, 2 phases, hard job, fine. 3 phases, okay, fine, okay, some hard thing, PhD, so 4, 5, 6, oh no, we can't do anything much. We can write down in-principle equations, but it's extremely difficult to solve anything. And living systems, as I will show you some pictures, are really in phases with many levels. So one would like to ask questions. So if I put a mixture of such phases, what are the emergent properties? How do their mechanical deformations look like? How do they respond to perturbations? What are the chemical compounds? Let's look at some pictures. I'm sure everybody recognizes this picture, right? This is a bacteria. This is an E. coli. Not a real picture. It's a very nice rendering by David Goodsell. I strongly recommend this book for a picture, for an introduction to biology. So this is a picture of E. coli. And so what Goodsell has done is drawn a very nice picture. And now we'll zoom in. Again, pictures and just hand-drawn pictures, but based on very realistic data. So you see the large scale stuff. This is about 2 microns in length and about 1 micron in width. And there are a bunch of strand-like objects which are coming. These are the flagella. These are the things that the creature uses to swim around. So let's zoom in. So there is this long yellow strand-like object. What is this? That's the DNA. This is really a polymeric structure which is already sitting there in the center of the cell, and that's the guy that does the job for the whole cell. Let's zoom in. So if you zoom in, you start seeing structures at various messy levels. This is long strand of DNA. There are lots of things appearing. There's a double-layered membrane. There are things sticking out of the membrane, and there is a end of a flagellum which is attached here. That's the motor which drives the object. Let's zoom in further. So there's a motor here which is really turning, and the turning of the motor is really what makes the object move. But you see lots of structures coming. There are ribosomes, there are free-floating mRNA, there's DNA wrapped around in messy proteins. Zoom in further and zoom in further and zoom in further. And then you start recognizing some of the things for which we can do physics. Simple water molecules, simple phosphate ions, chlorides, ATP, and so on. So this is your oxygen, two hydrogens and one oxygen. See the scale at which things happen and the complexity of putting many things together and trying to understand how the emergent properties. This is a pretty hard problem. We can at least write down something in principle if the system were at equilibrium. If the system were at equilibrium, would it be a living system? It would be dead, right? By definition. So we don't even know how to do the statistical mechanics of things which are out of equilibrium, even for simple things. But here's the things which are really, really complicated. So let's go back and zoom in there. So we go from there to that, there, and there, and there. And remember, this is possibly one of the simplest possible living creatures. We're nowhere near multicellular creatures. It's probably one of the simplest things. If you just put a bag of membrane, put some chemicals in there, and supply energy, this creature can suddenly move around. And that's bewildering to physicists. How is that possible to do that around? But this happens every day, right inside your gut, every one of us. Right? So you really look at the dynamics of the E. coli. So there's this long strand of flagella. So the cylindrical shaped object is the body of the E. coli. And you see that it does this fantastic thing called run and tumble. So now the discussion groups, we had a discussion about how the E. coli processes information by trying to move around in some medium. So what it does is tries to bundle up the flagella and rotates them counterclockwise. This counterclockwise motion makes it move in a straight line. It just doesn't keep moving in a straight line. Every now and then, it sort of unfurls one of the flagella and then suddenly turns around in some random direction, picks up the direction, and then keeps moving straight. And this process repeats on and on. So try to see if you can capture those dynamics here. So if you see one of the flagella opening up, this one for example, there you go, comes back again. This one, this one, comes up, opens the flagella and turns the direction. Somehow, if you put this in a medium which has a gradient of some food-like thing that you want, some food that it likes, this guy can read the information from the outside, process it, do all the stuff inside, make the proteins, generate the correct things, turn the motor around, and then move around. From a physicist's point of view, this is bewildering. How is this even possible? Remember the time scales on which things are happening. The chemistry that we do is at the level of microseconds. These are enzymatic reactions. There are ion channel gatings which really read the information from receptors and so on. It processes some proteins, it makes up those proteins, and then the proteins turn the thing, and the emergent behavior is what you see. This is really a bunch of objects put together in some compartment, and this is really a property of the entire thing, right? It's not a property of a single molecule. It's not a property of a single structure. So this movement, or whatever the characteristics of life, is really something which comes because you're looking at the emergent phenomenon. So when you think about living systems, you basically try to take point particles, spheres, polymers, membranes, put them all together, and like Zorana showed, put them in a mixer and churn them around, and out pops the thing. We don't know how. Not only that, if you look at higher organisms, they self-organize. I mean, this is again bewildering. It's not just at the level of single cells, but even many cells put together, like we have had many discussions on developmental biology and multicellularity. They really put themselves together and give rise to structures at very large scale. So this is a zebrafish embryo starting from a few minutes after post-fertilization. And if you just watch it, in about 1,000 minutes, it really builds an organism. So the things that I try to look at is how are these emergent structures, the periodic pattern of the vertebrae coming up. The vertebrae, and what sets the periodicity of that? How is the eye pattern coming up? How is it reading the information to make these patterns? They self-organize. That's the key word we use. That is, they put themselves together. So I keep emphasizing this term because the way putting themselves together is really what generates the different phases of matter. So in a sense, I try to look at living systems as particular phases of matter, which are driven by flux, We don't know how to describe them, but it's because you put them in a certain phase, characterized by certain ordering, certain dynamics, and certain interactions, that somehow these properties just pop up. It's a bit of a provocative slide, but I like to show it, and I'm happy to discuss that for a few minutes, but not too long. I want to do something else. This is from a book by Paul Alfred Weiss, Dynamics of Development, and he says the following, something which I really like a lot. He says, take a 6-day-old embryo, it's alive, it's in a jar, and you just crush it. Just kill it. Don't do much more than that. Just kill it. No change in chemistry. Don't crush it so hard you break the molecules. Just kill it. Just enough to kill it. Okay? So you can crush it. And then let's say you fractionate it. So you have put in a centrifuge and something like that, and then you get the heavier material at the bottom, lighter one at the top. So you recognize that the first one is ordered. In some sense, we don't know how it is ordered. It's ordered. And that's why it's alive. The second one is homogeneous. It's disordered because you crushed completely. The third one is also ordered, right? Because, I mean, you just put it in a centrifuge, and so the heavier ones came at the bottom and the lighter ones at the top. Somehow, this is not alive. This is alive. The same chemistry, the same interaction between them, but this was put together differently compared to this. So it's identical chemical composition. There's no difference between them. The other thing you know also is the following. One way is very easy. I can just kill the chick. But if I give you this jar, how do you make the chick? That's a pretty hard problem. Putting them together is extremely hard. So this is the question that I raised at the beginning. One is to describe what are the things that the chick is made of. Okay, I'll give you everything that the chick is made of. Build me a chick. That's much, much, much harder. Okay, can I go ahead? The big picture one tries to understand when you're trying to look at statistical mechanics is the following, that you have structure at various levels and they're interacting. There are genes, protein cells, and tissues. And the interaction is known to a good extent in the level of genes and proteins. But at the higher level, we really don't know much. In a StatMech language, there's a nice analogy. So there are interacting particles. You put them, a huge number of them. There's a machinery called statistical mechanics or thermodynamics, which lets us build useful things. We've mastered this sometime back in the last century. So we can do very, very good things. In biology, we know the interactions, and then there are organisms. We don't know what is the thing that really takes these interactions and makes organisms. So this is really from a book by Martinez Arias and Stewart. So I really encourage you to look at this. Really, really a deep book about development and from a physics perspective. So this is the real trouble. So we don't even know how to do the non-equilibrium statistical mechanics. But in the case of living systems, you have added complexity of the interactions. So we know what's here and we know what's there. We don't know what's here. We don't. In principle, also we don't know. So that's the trouble that we have. All right. So this is my last pedagogic slide, and then I'll go into some calculations about it. I really like this book, which sort of put things in perspective about what is it that we know and how did we know what we know. So Stewart says, Ian Stewart, who's a mathematician, there have been major 6 revolutions in biology. We understood how to look at small things, we understood how to classify them, and we understood why is it that there are a huge number of diversity, and we understood where that comes from, and the essential molecule that contains the secret of heredity. The final resolution is trying to understand how to put all these things together in a quantitative manner, and not just describing things in a human English language, but trying to put them together in a language where you can measure things, you can quantify things, you can build theories, and you can test theories. And this is what is physics and engineering done for us. All right. So with that, I'll switch to something definite. That was the general philosophy. I didn't want to go too much into that. So I wanted to talk about stochasticity in chemical reactions. So the plan is the following. I'll talk about reactions, how to describe them using stochastic ideas, and how is it that stochasticity gets averaged out, and does it always do that? And then finally try to think about stochasticity in spatial process. I'll come to diffusion. And towards the end of the lecture, I'll try to formulate a way to think about if you have things that are interacting via reactions and diffusing because of Brownian motion, they can lead to non-trivial patterns. And with this setup, tomorrow I'll bring in mechanics. How is it that mechanics can couple to patterns that you get in reaction diffusion systems? And those are the kind of things that I work on. stochasticity in chemical reaction. So I will not go into details of this. I'm happy to discuss this in one of the discussion groups. Let's try to understand. Say I have one molecule A, just one kind of molecule. Start with the simplest possible thing. And it's decaying. This is some unimportant, uninteresting product. It goes into something which I'm not bothered about. So the molecule is not being produced. There's a certain number of molecules of A, so let's say N molecules to begin with, and it's just decaying. And we say it decays with a certain rate, let's say gamma. Okay, is the formalism clear? Question is the following. If you just watch the molecules, does the number N go down smoothly with time, or is it really a stochastic function of time? Is the question clear? I have, let's say, 10,000 molecules of A, or 1,000 molecules of A. I prepare them in a jar, and I watch how many molecules are there at any point of time, and record that as a function of time. You'll get some trajectory. Suppose I repeat the experiment. I take another beaker, same 10,000 molecules, look at the number of molecules, watch them as a function. Will you get the same trajectory of the way the molecules will decay with time? Yes, no. It will be stochastic. Why is it stochastic? There's only one reaction. Let it come. Sorry. There's only one reaction. So she's saying it really depends on which reaction happened when, and so on. That's the stochasticity that is. There's only one reaction. Is there a stochasticity even in a single reaction? It depends on the time scale. Time scale of what compared to what? And the rate of reaction compared to? Okay. So there is an observation time scale and there's a reaction rate time scale, and she's saying it really depends on the two time scales that you're looking at. That's correct. So we'll come back to that. There's also the fact that if the number of molecules is small, again, ask questions about how small is small. Small compared to how much you can detect. If there are 10 to the 23 molecules, I mean, you can't detect 1 in 10 to the 20. It's impossible. If there are just about 1,000, maybe 1 or 2 you can detect. 1 or 2% of variation. The basic idea, like you all pointed out, way the chemical reactions proceed is stochastic. There are lots of effects around molecules, they come. You don't know when each guy decays. If a guy starts from A, it will become something phi, some intransition. But when? At what point of time? And so therefore there is a stochasticity in how many molecules are there at any point of time. So if there is intrinsic stochasticity, we would like to describe this dynamics by a probability function. So the probability is the following. So what is the probability? The function of time of finding n molecules of A remaining in the jar. The experiment is that I have a jar, I have a certain number of molecules of A, I count the number of molecules of A, and then I look at the time course of A. What do you call? Yeah. So if you look at this once, you might get something like this. If you do the same experiment again, you might get something like that, and so on and so on. So therefore, I would like to ask, if I look at a single time point, some point of time t, what is the probability I will have little n molecules of A? Is the formulation clear with the model? So in particular, we would like to understand the following. So how does the probability P, N, change with time? Or how does the number of molecules change with time? How can the molecules change with time? Suppose you had n molecules. And if there's one reaction that goes on, it can become n minus 1. Is that right? There's one molecule decayed, and so the n changed to n minus 1. But you could also have been in a state n plus 1, and one molecule decayed, and you could come to the state with n molecules. So you can reach the state with n molecules, either by coming from a state n plus 1, or leave the state with n molecules by going to a state with n minus 1. Is that clear? So if you ask, how does this probability change with time? This is a way to build a mathematical model for the probability of the reactions. Remember, each reaction proceeds with a rate gamma. One can derive this from more elementary means by looking at each and every molecule and constructing averages. I can do that in a discussion group, but let me go ahead. So you could be in a state with n plus 1 molecules. This one. And one molecule decayed, and with a rate proportional to gamma. And which of the n molecules? Well, there were n plus 1 molecules, so each one of them could have decayed. And so the probability that one of them decayed will be proportional to the number of molecules. So you get a production. You could also go to the state from n to n minus 1, so you get a term there's no n there. Is this clear, the formulation of the model? If not, let's just spend some time on this, and then we can go on to the more complicated. Repeat what? Okay. So I'm trying to ask, there's a single reaction. It's intrinsically stochastic. So if I look at different realizations, there'll be a different number of molecules at every point of time. I'm trying to ask, what is the probability of finding n molecules at any given point of time? And I'm trying to ask, how does that probability change? Well, that probability can change because a reaction happened, and the reaction could have happened from n plus 1 molecules to n molecules, or the reaction could happen from n molecules to n minus 1 molecules. So there are two terms. You can come into a state with n molecules, either because you started from a state n plus 1 molecules and one molecule decayed. Or you could be in a state with n molecules and one molecule decayed, so you leave that state and go into n minus 1. And both of them have the same rate gamma. So if you had n molecules and you want to go into a state with n minus 1 molecules, how many ways can you do that? You could have this guy decay and the rest of them not decay. But you could have any other guy decay as well, right? So how many ways can you go from a state n to n minus 1? N ways. So I started from a state n plus 1. I'm going to a state n. How many ways can I do that? N plus 1. But if you're going from a state n to n minus 1, remember I'm interested in the state n, only one molecule should decay. Oh, my apologies. There is an n there. So that can also happen in n ways. You're right, sorry. I put a subscript. That's a stochastic description. But you know, on the average, what happens intuitively. So if you just count the number of molecules on the average, look at many, many realizations, count the average number of molecules, how should the average number of molecules change with time? Will it go up? Will it go down? First thing. Or will it remain same? So I start with capital N molecules. I do many realizations. Let's say I have 10,000 flasks. And each flask I count the number of molecules. And I look at the average of them. Remember, they're all just decaying. They're doing nothing else. So what will happen to the average number of molecules with time? It will go down. How will it go down? I heard the word exponentially. Yeah. How does it go down? You know? How does it go down? Suppose you didn't know about all this stochastic kinetics and blah, blah, blah. So you were just saying that I have a master equation, not master, the law of mass action with a rate A. So how does the concentration of A change with time? Minus gamma. Is that right? This you know how to solve. So it will just go down exponentially with time. In fact, if you look at the average, that's exactly what it does. However, if you look at individual trajectories, they'll go down stochastically. I started with something very, very trivial where it's obvious that if you just take the average of a large number of them, you'll get the behavior that you expect from a deterministic differential equation. Nothing surprising. So I urge you to do one more example. I will not work this out in detail, but just one simple complexity. So let's say the molecule A gets decayed at a rate phi, goes to phi at a rate gamma, but is also produced by some other reaction. I don't know what those reactions, I don't care. So it's being produced at a certain rate K, and it's decaying at a certain rate gamma. So can you tell me what this should look like? How does the concentration of A change with time? I'll write down one term, you write down the other term. I'll write down one term. You write down the other term. Plus K. Is it clear? Why is it plus K? So there'll be a production at a rate given by K. So again, if you do this experiment many a times in a system with a small number of particles, you'll get stochastic fluctuations. So again, tell me how does average and change with time? This time, will it go to 0 at long times? Will it do something finite? What should happen at long times? So at long times, the system, you can say it reaches some sort of a steady state. Just set the rate of change to 0. And so what is the concentration at long times? No, it goes to K by gamma. So this will reach a value which is K by gamma. So if you start with 0 molecules, it will go do something and then it will finally reach. The same thing, if you look at this many realizations, there will be stochastic effects coming all along. Each color is a realization, the white line is an average. So again, in this case, there's no problem. I mean, the average behavior is just, or the deterministic behavior is just the average of the stochastic behavior. There's no problem at all. And with the same rates, that's important. So whatever the rates you thought were occurring at the microscopic level between the chemical reactions are the same rates that will appear at the deterministic level as well. So now let's complicate things a bit and see where is it that this kind of a distinct picture breaks down. Suppose you had a nonlinear reaction. That means I have two molecules A and B. They interact and give you a molecule C. And this goes with a rate F. And the molecule C decays with a rate B. Okay? And I just ask you, tell me how does the concentration of C change with time? Simple law of mass action kinetics. How do you write that? So there will be the concentration of A multiplied by the concentration of B by F. I'll put a F tilde, okay, and then minus D and into C. I'll put a tilde. Question is the following: is F tilde and C tilde the same as F and C? So not C, D tilde. Are they the same as the microscopic reaction rates that you put in, or are they different? So in these two cases, where there was a simple degradation or a simple degradation followed by a production, the rates that you get at the deterministic level are the same as what you put at the stochastic level when you average over the stochastics. In this case, you have a nonlinear reaction in the sense that the rate of change of C depends both on the level of A and the level of B. And at the microscopic level, the rate constant is F. If you do a simple mass action kinetics, you would say, okay, it's the same F. So if you have a large number of molecules, again, with the order of, say, Avogadro number, then it is true that the reaction rate at the level of the deterministic description just scales with the volume of the system. And the, okay. However, for small n, where fluctuations are dominant, they are not negligible. F tilde is not equal to F times. However, the rate D tilde is always equal to D for all n. So the rate at which the D was decaying, or the rate at which C was decaying, that was a simple linear reaction. And so if you add up all the stochasticities, they just add up. And you get the average, and that looks like the macroscopic deterministic behavior. However, if you have nonlinear reactions, what you will do by just looking at the average behavior is that you'll be missing out the correlations that are present between A and B. In the following sense, if I ask you for the average of the product of A and B, is it the same as the average of A into average of B? Let's say A and B are two random variables. Is average AB the same as average A into average B? If A and B are independent random variables, then it's true, but not otherwise. And surely here they're not. They're reacting with each other. There are correlations between them. And so if you take nonlinear chemical reactions at the microscopic level, look at their deterministic descriptions, you miss out the correlations which are there if the number of molecules is small. And so the rates that you will put in will have some correction terms which you'll usually miss out. Okay, so that's all I wanted to say about stochasticity in chemical reactions. So this is stochasticity in just looking at chemical reactions, right? So there are molecules and they're interacting with each other, or maybe they're just dying down. And those things itself are stochastic. And if you really look at their deterministic dynamics, or you look at only the deterministic limit, you will miss out some of the really important correlations that exist in these systems. So what I really wanted to focus on are stochasticity in the way the molecules move around as well. So just to make connections with other talks, what I'm thinking about is a well-mixed system. That means there are no spatial profiles of A. Everywhere it's the same number of molecules of A on the average. And so I'm just looking at how they decay in time. Surely the molecules are also moving around. They diffuse around. And so therefore there can be stochasticity in the way they move around. And the way the stochasticity in intrinsic chemical kinetics via reactions and the stochasticity in the way they move around in space, diffusion, the way these two things couple is what leads to reaction diffusion patterns. And what's the basic cause for diffusion? Why do you get diffusion? You understand diffusion, right, everybody? Let's try to understand where is it that diffusion comes from, actually. So diffusion really comes from Brownian motion, right? Everybody must have heard of Brownian motion. So if you look at pollen grains, that's what brown bread, just put them in some solution, just observe them in a microscope, they jiggle around a lot. They don't stay in one place. What's the microscopic reason for that? Why are they doing that? Yeah, so there are plenty of other tiny molecules which are unable to see, and they are really hitting them around. This is not to say they don't obey Newton's laws. Newton's laws are still valid. It's the same physics that we learn in high school, F equal to ma. It's just that you're not looking at all the molecules. You're only looking at the molecules that you're interested in. But the other molecules that you're not looking at will come into play. They will hit you around. And the effective interaction that turns out between these tiny molecules and the large one is what you see as Brownian. So we can make it more quantitative from here on. So that's really what's the picture. So there's one molecule that you're looking at, and the trajectory of the molecule looks like a random walk. It's a trouble. I mean, how can things be random? Didn't Newton say F equal to ma? Where is the randomness there? Oh, there isn't. The randomness is effective in the way that you're looking at. It's only a reduced description. You're only looking at the green guy, forgetting all the gray guys. And so therefore, the effective dynamics of the red guy, green guy, looks like it's stochastic. So we can write down this very, very quantitatively. So we can write down Newton's laws, rate of change of momentum, mass into velocity dot, equal to the total force. So if you have the force, the equation of motion for the green guy, the total force on the green guy, turns out you can have two parts. One is there are some external forces, say gravity. This we know how to do from school, right? Half GT square, and everybody knows it by heart. But there's also the part that comes because it's colliding with the other molecules and you're not looking at their dynamics. This collision force itself, two parts. One, there is the average deterministic force. Again, something that we've all done in school. And there's a part which comes by the fluctuation, the stochasticity, the part that you're not keeping track of, the part that where you don't worry about the positions and velocities of all the tiny particles in the system. The average force is something simple. We all do this in school. Take a steel ball, drop it in a glass of water or some oil, and you calculate the terminal velocity. You calculate the frictional drag term. The same frictional drag term, 6 pi eta AV, that comes. There's a part that comes by looking at the stochastic forces, the random forces, so that we don't know how to describe, and we say that, okay, they are drawn from a probability distribution. On the average, it has nothing, no value, its average is 0, but there are some small correlations. And if I look at time scales longer than those small correlations, it really looks like uncorrelated fluctuations in the system. So that's how you build what is called a Langevin equation description. This is really just Newton's second law. How do you get diffusion from here? So one could look at the following picture. Just like we do Newton's laws in an overdamped limit, that is, we look at time scales where inertia is negligible and it's really friction that dominates. We can look at the same the Langevin equation at long time scales, and if you do a bit of math, I'll not go into the details, we can talk about more, you'll get what is called a simple random walk model. The random walk model just says there's a particle and it just takes random steps at every instant of time. That's the Brownian motion picture that Andras showed. It just takes random steps every instant of time. This can be clearly derived from just Newton's laws for the entire system and looking at the dynamics of only one of the particles that you're interested in. So there's one thing that I wrote down. So somehow the strength of the noise, the strength of the stochasticity is related to the diffusion coefficient. How is that? We'll see this. Let me skip this part and I'll come back to this. So how do you think about diffusion, the classical picture of diffusion that we all think about? There are a bunch of molecules. You create a local perturbation. Let's say you put more molecules here and less molecules here. What will happen? The molecules will diffuse around. Take a drop of ink, put them in water, it just diffuses around. So it's really gradients in the concentration, variations in the concentration profile that will drive currents, that will drive diffusive currents. Right? That's the equation that we all write down somewhere here. Here, the diffusion equation. So we say the rate at which the concentration in a particular region changes is just proportional to the second derivative of the concentration itself, the second derivative in space. I just want to emphasize that if you look at the Langevin equation and the random walk model that I wrote down and try to construct the concentration profile, you can clearly derive that this again, the concentration profile really satisfies the diffusion equation that you know. So let's now go back to the part where we look at stochasticity in space and try to see if you can make that into a mathematical model. So we started from Brownian motion. So this we said is just the random, apparently random dynamics of any particle in a fluid. It starts from here and goes around in a random fashion. And this, in a simple language, we can quantify it by the random walk model. So the random walk model just says that take a particle, add a random displacement to it in any direction, and just keep doing that process. And the trajectory that you will generate is typical of the trajectories that you observe for a Brownian particle in a fluid. Again, we try to construct a deterministic description This is a stochastic thing. By talking about what we call diffusion equation or the diffusion process. So what's the diffusion process? It says that if you have C, let's say, which is the concentration of the molecules or the number density of the molecules that you're looking at, and ask how does C change with time. So in a one-dimensional scenario, just for simplicity, if you have particles which are sitting on this lattice, let's say in this room, and I'm just standing here, and I do a random walk. So I just randomly take a step to the left or to the right, and I keep repeating it. I don't keep doing that, but I keep doing it apart. So that's the stochastic description. But suppose you do this many times, and then ask, what is the number of molecules, or essentially the concentration at any point of time? How does that change with time? So that's essentially the same thing as describing what is the probability of finding a molecule at a particular place in space at a particular point in time. This you know the following, right? So if you have a region with a more number of particles here, say Na, and you have less number of particles here, Nb, in the region A, and where will the particles move on the average? From left to right or right to left? Left to right. Why? You said something about equilibrium. I don't even want equilibrium for this to happen, but okay. Yeah. So he's saying basically the particles can do a random walk independently going to the left or the right, but the number of particles on the left is more compared to the one on the right. And so therefore the number of ways from going to the left is much more compared to the number of ways of going to the right. So on the average, the particles will move from left to right. Sorry, Ram. Yeah, I was going to derive that, but so that's essentially what Ramray said. It's fixed law. So what really matters is the gradient in the concentration. It's the way the concentration changes with space. That tells you what is the current of particles that are coming into the system. And fixed law is just saying that the current of particles is directly proportional to the gradient of the concentration. But I wanted to ask you, how does the concentration field change with time? Surely you're not creating or destroying particles, right? I mean, the total number of particles is constant. So if the particles move from here to there, the particle number, if you integrate over the entire system, didn't change. What changed was only where the particle was. And so therefore, the concentration field should obey what is called a continuity equation. Again, this can be very easily derived. I strongly urge you to do that. Just by looking at how particles move from here to there and there to here, and you will find that the spatial gradient of the current is what leads to the rate of change of the concentration. So this is what is called fixed law. This is the continuity equation. So if you put fixed law inside the continuity equation, what do you get? By the way, I'll use subscript for derivative. What do you get for the time derivative of C? Just have to put this definition inside here, that's all. You get the diffusion equation. Is that clear? So stochasticity in the way the particles move around comes by because you're neglecting some particles and looking at only few of them, leads to the random walk model. And the same random walk model, when you just look at it in a coarse-grained description, try to write down a deterministic description, you define some averaged quantity called a concentration, and see how the concentration changes with space and time, and you get to the diffusion. The diffusion equation is what is called a partial differential equation. Partial because C depends on both space and time, and so the derivatives can be either with respect to space or with respect to time, and they're both coupled, and the coupling is what gives rise to the law. Really running out of time. So how do we solve the diffusion equation? So I'll not solve the equation mathematically, but you can get it out from a simple physical intuition. You know how to do this intuitively. Suppose I have this region, let's say a bounded region, and this is my coordinate x, and I create a concentration profile which is like that. How does the concentration change with time? What will happen at long times? Will the concentration be some profile? Will it be flat? Will it go up? Will it go down? Everybody is cutting. Yeah. So at long times, so this is the initial condition. Long times, you all know intuitively that this will become something which is flat. Is that right? One can look at the solution of that. So you can just take in this equation, put it in here and solve it. It's a simple mathematical calculation, but I'll not go through that. So you know that this is how it looks like if you wait long enough. I would like to ask the following. I would like to ask the stability of the uniform state. In other words, I take the uniform state, C as a function of x, and I say I put a small perturbation. And then ask how does this perturbation change with time. Remember, the concentration field is obeying the diffusion equation, and I ask how does this perturbation change with time. What happens? Will it go back to the homogeneous state? Will the perturbation remain? Will the perturbation grow? Huh? It will go back to the homogeneous state. Sure? Everybody understands that? Suppose I put a perturbation in the other direction, then? Huh? It's the same. Does it depend on where I put the perturbation? No. So wherever I put the perturbation, and no matter what the perturbation I put, the system will just go back to the flat uniform state. So therefore, the flat uniform state is a stable state of the diffusion equation. Now, if you have a region with a bounded domain, in other words, it's a finite size domain, let's say of size L. Think of this as some container in which you're looking at the reaction. And you have a buffer where you maintain the concentration of the molecules at some level on this side and some other level on this side. So let's say here you maintain it to be C0, and here you maintain it to be C1. Remember, this is a partial differential equation, or like any other differential equation. You solve the equation, but then you have to fix some boundary condition, like you do initial condition. Here will be boundary conditions if you look at the steady state. So I want to look at the steady state solution. That means I set dTc equal to 0, which means my d times this equal to 0. And I want C to be equal to C1, or C at x equal to 0 to be C1. And I want C at x equal to L to be equal to C0 and C1. Can you solve this differential equation? What is the solution of this differential equation? Second derivative equal to 0. Is it too elementary or too advanced? I'm not getting any feedback. If it's too elementary, I can go up. If it's too advanced, I can go down. Just tell me something. Very good. You don't understand what? You don't understand what the setup is. Okay, fine. But you understand the differential equation. Okay. So I have a pipe in which, let's say, I put in some liquid, there's some water thing, and I have some dye. And I'm trying to look at how the dye profile looks like in the pipe. One end of the pipe, I maintain the dye concentration at some value C0 by some buffer. The other end of the pipe, I maintain the dye concentration at some value C1, again, a different buffer. Question is, I leave the system forever, long time. What is the concentration profile as a function of space? Is the setup clear? And we derived that the concentration obeys the diffusion equation. I'm just saying you, try to solve the diffusion equation subject to these two conditions, that one end the concentration is fixed at C0, the other end the concentration is fixed at C1. And you're looking at only the long time, steady state limit. So the time derivative goes to 0, long time, it doesn't change with time. So I'm just trying to solve a differential equation where the second derivative of C is 0. And C at 0 is C0, and C at L is C1. What is the solution of this differential equation? It's linear, just a straight line. So C of x will be your usual mx plus c. No, not that c, x plus d. You can fix your c and d, m and d, the slope and the intercept, just like you do in school, by demanding that c of 0, which means that x equal to 0 equal to d should be equal to c0, and c at L should be m times L plus d, and that should be equal to c1. So d equal to c0, can you figure out what is m? So it's c1 minus d divided by L, and so therefore the concentration profile looks like c1 minus d divided by L times x plus why am I doing this? So if you look at Drosophila, you all know in the very early embryonic stage, at least the biologists know, so there's this object. There is a certain molecule. Yes? Oh, yeah, you're right. And D is equal to C. So if you look at Drosophila in the early stage, you know that the mother does something and put some molecules of mRNA somewhere in there, and this molecule expresses some protein, and there's this concentration profile of bicoid that comes up. So this is the famous concept that you all know, is morphogen gradients. So there is a certain morphogen, in this case it's bicoid, and there is a gradient of morphogen. And the gradient of morphogen is really what is apparently trying to set up the pattern. How? I'll show you probably tomorrow. So there's a very nice paper by Francis Crick, I don't remember the year, 60s maybe or 70s, where he tries to explain this morphogen gradient in Drosophila by appealing to this picture, that maybe there are two buffers and you're just trying to satisfy the solution of the diffusion equation by matching the two buffer condition, and that therefore will give rise to a profile of C, which will just be a straight line. So where it will be C0 at one end and C1 at the other end. Turns out this is not true. And the molecules are not just diffusing, they also do some reactions. So they are being produced at a certain rate, or they are being buffered at a certain rate in one end, but they also decay, and that's what really sets up the gradients. So remember what we have been trying to do. Looked at stochasticity in chemical reactions, and when is it that you can neglect them. Then we had stochasticity in the way the molecules move around. And if you try to look at the deterministic behavior, deterministic description, what you get is the diffusion equation. And what we have just seen is that if you don't have any boundary conditions imposed, the homogeneous state is a stable, steady state. On the other hand, if you have two boundary conditions via some buffering imposed, then you can have a non-trivial profile of the concentration. So that's the concept of the morphogen gradients, right? No. So as like I was saying, there's bicoid in Drosophila embryos. Bicoid turns out to be expressed only at one end of the embryo, and the way it decays down, the other cells or whatever the proteins that are there, they read the concentration profiles and do something else. That's a standard French flag model developed by Louis Wolpert in the late 60s. Now, like I was saying, it's not just the diffusion equation. Turns out this is the first system where we start looking at the competition between reaction and diffusion, and they both set up a very nice pattern. So I'll solve that and then I can stop. The molecules, whatever, let's say use B for bicoid. So there seem to be two things that's happening to bicoid. One, it diffuses. Can I take another 5 minutes? Allow the diffusion equation. But it also does some reactions. Let's just use the simplest reaction that we learned, linear degradation, and nothing else. So the molecule B decays at a rate K. And if you just look at the deterministic level, how do you write the concentration of B, the equation for the concentration of B? You know that it is just K times B. Yeah? In addition, I'm saying that it diffuses. And a boundary condition that B equal to some value B naught at x equal to 0. So for simplicity, you can think of Drosophila as being an infinite-sized embryo, where it has a boundary at one end, x equal to 0, and the other end there's no boundary, it's unbound. So the equation that I've written there is another way of imposing the boundary condition by saying that there is a production term at x equal to 0. For those of you who know, it's a Dirac delta. For those of you who don't know, it doesn't matter. If you try to solve this equation, remember this is as a function of space, not time, and look at the solution of the differential equation at long times, the steady state, you'll find that this is really an exponential pattern going like e to the minus lambda times x, where lambda I just wanted to point out that this is not just a theoretical fancy. If you really measure the concentration profile of bicoid, it really matches very well to an exponential distribution. I urge you to look at the work of Tom Gregor. I didn't get the plot with the concentration profiles mapped onto the theory, but this gives you a very nice explanation. Not only at the early stages, if you look at the patterning of the wing disk in Drosophila at a later time point, the patterning of the front and the back of the wing, the posterior, okay, whatever, distal and proximal, those regions, turns out there's a region of the wing disk which expresses a certain protein called DPP, decapentaplegic. And this protein again decays at a certain rate K and is being produced at a certain position and also diffuses. So you can measure the concentration profiles across the wing disk. That's the jagged line. You can also calculate the solution by a diffusion-like argument. And there is quantitative matching between the numbers that you measure in the experiment and the profile that you calculated here. Yes. Where is the third term? This one. So this is a way of imposing the boundary condition that I said, that you fix the concentration at some region equal to some value. So that B naught turns out to be square root of J naught by K. It's a Dirac delta function if you know the math. Otherwise it doesn't matter. So what we are looking at is a situation where we are looking at the steady state profile. We're looking at a long time scale. And there is a certain length scale that comes up with the exponential decay. And that length scale is really the ratio of how fast you diffuse compared to how fast you degrade. So D is the diffusion coefficient, K is the rate of degradation, and the ratio of the two sets a length scale on which the profile decays exponentially. I'm looking at the long time steady state. You can easily work out the full time-dependent dynamics of the system, and how the system will approach the steady state will of course depend on what is faster. It is diffusion faster, and so on. Okay. So I'll just flash through this. This has been discussed a lot, but I will not work out any details. So this is a system which gives you some sort of a non-trivial profile or patterning because you impose certain boundary conditions on the problem. Turing showed that if you have two such species which are interacting, F and G are some chemical reactions, may not be exactly whatever, I mean, there's some general structure to the reactions and not specific terms, and they diffuse. This inverted delta is just the second derivative operator, just done in many dimensions. Turing showed that if you have two such species, which they are interacting with each other, and if there was certain chemical topology, I mean, one of them is an autocatalytic term, there's an inhibitor, which also tries to reduce the activator tries to make more of the inhibitor, and if the two species diffuse differently, I think the activator should diffuse faster. I always mess up this. Activator is faster, yeah. Inhibitor is faster, yeah, I'm not going fast. If the inhibitor diffuses faster, then Turing showed that this system is spontaneously unstable to fluctuations. So I asked you the following. If you look at the diffusion equation, and you look at some small perturbations to the diffusion equation, you all agreed that it just decays, no matter what the perturbation I put. But I said diffusion equation plus reactions, and now put a small perturbation, and you can ask, does it go back to the homogeneous state? Turns out no. If you satisfy some criteria, which is the Turing criteria, activator, inhibitor, activator diffuses slowly compared to the inhibitor, then this system can spontaneously lead to non-trivial patterns. Well, this was a theoretical speculation for a very, very long time, but recent works, I've not put in everybody, I've just put in a few of them, so apologies to those who I'm not acknowledging. Very recent work by James Sharp and company have shown that this is really true in the way vegetation patterns come up in the mammalian limb. Many other examples of hair follicles, turtle shell patterning, and so on and so forth, where one doesn't just try to understand, oh, is it up and down and nice patterns, but one can actually measure numbers and try to see they satisfy the Turing criteria and maybe try to predict the wavelengths of the pattern that come. So again, I'll just flash the equation. I will not do this. There is a way to quantify the statement by asking, is it stable? Is the stationary state stable against small fluctuations? This is exactly the kind of analysis that Sandeep did last week for an ODE system, but you can generalize this to a PDE system. You look at the state, put a small perturbation, ask whether it comes back or not. So like Sandeep said, you look at the Jacobian matrix, and then in this case, it's not just the Jacobian matrix, it's the Jacobian matrix plus the diffusion term that comes up. And because it's really diffusion and reaction, some sort of a combination comes up, and that gives rise to patterns. I'll not go into the details, I'll just show some nice patterns and I'll stop. So one can cook up lots of nice models of the chemical reactions. One favorite is something called the BRUSSELATOR. You can probably build something, many other molecules as well. Maybe you can take Sandeep and Sunil's thing and make it Sandeepator or Sunilator or Sandeep Sunilator or Sunil Sandeepator. Okay. Anyway, so this model was done in Brussels, so it's called BRUSSELATOR, that's it, nothing else. There's an oreganator. And so one can do the linear perturbation analysis, and then it turns out that certain parameter regions, they do start giving patterns. I'll just show you the phase diagrams and I'll stop. So that's the phase diagram. So if you look at the space of parameters, the diffusion coefficient of one of the molecules, the reaction rate of some other molecule, there are regions of the phase diagram where the homogeneous state is stable. That means if I look at the homogeneous state, put a small perturbation, run the diffusion equation, the system just comes back to the homogeneous state. Nothing doing. The homogeneous state is stable. There are regions where the homogeneous state is unstable, but it oscillates. This is the spatial analog of what Sandeep did. So there you had ODE where it was oscillating in time, where it's homogeneous in space, but is oscillating in time. There's no pattern in space, but there's a pattern in time. And finally, if you satisfy the Turing criteria, you can get patterns which will settle down into a nice periodic pattern. So these are the standard Turing patterns there. Okay. So let's just summarize from this nice paper. You can have one molecule, morphogen. You specify some boundary condition, like B0 here, and that can give rise to some profile. You can use that profile to do some patterning on your system, make some self-weight determination or something like that. I'm not going to go into the details. Of course, you can have it upside down. You can take the French flag and work. Or you could have two of them and maybe put boundary conditions one way this and one way that and play with all possible combinations. You can start getting probably all country flags. The other one is where there are no boundary conditions imposed per se, but there are interactions autonomously occurring between the species. And you satisfy certain criteria, which you can derive very easily by doing the math, and they lead to spontaneous patterns in the system. These are the standard Turing patterns. So tomorrow I'll tell you about this story. So I have worked on this problem where there's a very nice connection between pattern formation and the kind of mechanics that Olivier talked about last week. So I'll try to describe a pattern that emerges in the surface of certain cells, C. elegans polarity. This pattern is very important because it also establishes developmentally the anterior-posterior axis, the head-tail axis of the worm. This pattern is interesting because it turns out that there are complexes, the blue complexes and the red complexes, which I call them A and P collectively. They have mutual phosphorylation among the complexes. This mutual phosphorylation leads to antagonistic interactions between the A and P. And as soon as you have this kind of a picture, you should remember from Sandeep's discussion, this leads to bistability. So the bistability is what leads you to go from a domain like this to a polar domain, and so you establish an axis from the red to the blue, and therefore you establish the anterior-posterior. So this problem will have connections between biochemistry, as evident here, and diffusion, and also the mechanics of the actomyosin cortex, which I'll tell you tomorrow. I'll stop there.
Time for one or two questions. Okay, so I have one. I try to connect your two parts of your presentation. So in the first part, you said that maybe we cannot explain biological system using the typical physics approach, supposing that the system is at equilibrium.
Yeah.
But correct me if I'm wrong, but in this part, you show that we can actually use kind of physical formulas using assumption of...
No, that's not right. Of course.
Okay, sorry.
Just being arrogant, biology is physics. But independent of that, if you are at equilibrium, the way the diffusion constants are related to the material properties of the medium, the viscosity, for example, there are very nice physical relations. If you do not satisfy them, you're out of equilibrium. I did not tell you how to find out the diffusion constant. If you're at equilibrium, the diffusion constant of the molecule will be related to temperature and the viscosity of the medium. If you're not at equilibrium, there's no such relation. Temperature is an independent quantity, diffusion is an independent quantity. So the description that you try to build for time-dependent dynamics in equilibrium gives you particular things which we know. But you can also generalize that to non-equilibrium and so therefore time-dependent.
Yeah, thank you. In the case of the probability of the intrinsic stochasticity, do you know if there is some kind of a common distribution of this probability independently of chemical reaction? Of every chemical reaction has its own intrinsic.
So I wrote down an equation of motion for the probability itself, which was the master equation. So you can solve the master equation, and so therefore get the probability distribution. And the probability distribution will of course depend on the terms in the master equation, which will be dictated by the kind of chemical reactions that you have. The probability distribution will therefore depend on the kind of chemical reactions that you have.
Thanks.
Hello. Yeah. In living systems, you think if by adding more feedback loops, the stochasticity can be reduced and could
I don't think you can make a general statement like that. You might increase, you might reduce. I do not know.
No more questions? Okay. So thanks again, and we can go for lunch now. Thank you.